Hello, and welcome to Science Journeys, Caltech's first online science education series. I'm Michael Alexander, the Director of Caltech's Public Programming, and I'm very happy to have you all with us today. Caltech is one of the nation's premier science education and research institutions. In fact, Caltech created the Jet Propulsion Lab, JPL, which has managed most of the United States' unmanned space flights. As we speak, there is an unmanned space mission on its way to Mars that includes a helicopter that will be the very first to ever fly on another planet. Our office is also responsible for bringing to the campus the Performing Arts series that takes place here in Beckman Auditorium. We would encourage you all to look to our website, where you can find out about all of the lectures and performing arts series that are going on here by going to events.caltech.edu. I am really pleased to bring to you today Cecilia Sanders, a young scientist who has become a real hero in our office because of the exciting and informative talks she has presented as part of the series that we present to students in our auditorium. She shares information in a way that even non-scientists like me can find accessible. She mixes the study of living things and rocks, at least those living things that she can find in fossils. If you are interested in information about the first life on Earth, this is the talk for you. We have also asked all of the Science Journey lecturers to give you a bit of their early life story, so maybe some of you could figure out if science is part of your life plan. It is now with great pleasure that I introduce Cecilia Sanders.
Thank you, Michael, for that extremely kind introduction. Before we get started with this presentation I have for you all today, I've been asked to talk a little bit about my background and some of the things that have happened to me before I came to be on this stage right here. My name is Cecilia Sanders. I am a fourth-year graduate student in a PhD program in geobiology here at Caltech. So geobiology is the study of both geology, which is the study of rocks and minerals, and the study of biology, so the study of living things. So you can think of geobiology as the study of the interaction between rocks and minerals in the non-living world with the living world. I've also done some work on the problems related to planetary evolution. Planetary evolution being the study of everything that happens to a planet from when it's formed to the present. So that includes climate change, that includes tectonism, that includes the evolution of new clades and styles of life. But really, all of my research can be boiled down to the study of wriggly things on rocks. So squiggly lines and bumps and lumps form patterns and pictures that help us figure out what the world was like at the time a rock was formed. This helps us build the larger story of everything that's happened to a planet and that's happened to a particular environment through time. My email, in case you're interested in reaching out to me afterwards, or if you just want to send me some pictures of interesting rocks that you found, is csanders@caltech.edu, and I can put that up later. So nobody starts, nobody is born as a scientist. We're all kids first. This is a picture of me as a kid around the age of 3, fording the rushing white waters of a huge river somewhere in the Appalachians. When I was a kid, I wasn't what you might consider to be particularly academic or particularly studious, and who is before you really start grade school in earnest. I grew up on the East Coast with my mother, my father, my sister, and my grandmother, and then later my little brother. There he is, a little baby. And when I was small, I wouldn't say that I was especially interested in rocks, or especially interested in minerals, or especially interested even in paleontology. What I do remember of being 6 years old was being completely obsessed with tiny things. I was really interested in tiny creatures and tiny pictures and stories about tiny people. And I spent so much time and so many crayons and markers and pieces of construction paper creating for myself tiny dolls and tiny models. And as I was putting together this presentation and thinking about what are some of the connections between the person that I was when I was small and the person that I am now, I think that's probably key. I mean, my favorite toy when I was small was a little plastic horse that I got out of a vending machine at a grocery store, I'm pretty sure. It was about the size of my pinky nail, a little brown horse that I named Chocolate. And I loved to come up with stories about everything that's happened in Chocolate's life. And I wept piteously whenever I lost Chocolate. And now, as a geobiologist, a lot of my work involves looking at these small, intricate things and coming up with a story about them, trying to explain everything that's happened to them in the course of their lives. And so I think that the lesson here is not just that I was absolutely adorable, which is very clear from these pictures, but that just because you aren't mixing chemicals together and looking through a microscope when you're a tiny child doesn't mean that you can't become a scientist. I really passionately believe that the only two things that you need to be a scientist are creativity and curiosity. And every kid is full of creativity and curiosity. And the things that may not seem like they're helping you to become a scientist or an engineer or a professor or something right now at this second might end up being things that influence how you see the world and help shape your skills later on in life. So I was also asked to say a few words about my academic history, my education. So as I mentioned, currently I'm a PhD candidate in geobiology here at Caltech. But before that, I got my master's degree in planetary sciences, also here at Caltech. Before that, I was an undergraduate. I went to college at Harvard and got a joint degree in Earth and planetary sciences and astrophysics. But before that, I was a high school student, before that a middle school student, before that elementary school student, before that a preschooler. The schools that I went to were all public schools back again on the East Coast of the United States, with the exception of Bowie Montessori Children's House, where I went before the age of 7, and which, for all its eccentricities, I really do credit with my love of learning and being curious and finding creative ways to explore the world around me. Really, my early education was the time, I think, that all of those fundamental things that I think it really takes to be a researcher were really nurtured. So that is enough about me. And now I'll move on to the science part of our program. Let's talk science. So I titled this talk, Beyond Bones, Interrogating the Fossil Record of Small, Soft, Profoundly Earth-Shaping Organisms in the Precambrian. And we'll talk about what a lot of these words mean in a moment, but really I called it Beyond Bones because typically when people think about the word paleontology or the study of the history of life or ancient life on Earth, things like that, we picture something like this. This is an image of a part of the dinosaur hall at the Natural History Museum of Los Angeles County, which some of you may have been to. Dinosaurs are cool. Bones are cool. It's cool to see the skeletal remains of organisms that once walked the Earth. But that's not all there is to the story. If you've been to any natural history museum or if you've been to the beach, looked at some beach rock, you've also probably seen things kind of like this, maybe ammonites, maybe snails, clams, other shelly creatures, but both the shelly animals and the dinosaurs and the mammoths and all of these things, what they have in common is hard parts. These are hard-bodied organisms. And there's a reason that we picture them when we picture the whole field and study of paleontology, because these are the organisms that dominate most of the geologic record. But we know that looking at the world around us, not all of the living things in our world have hard parts. Not all of them have bones and teeth. Not all of them have shells. And a lot of them are made entirely up of biodegradable parts. That means parts that can rot and be eaten and can fall apart after the organism dies. So I made this sketch of just some examples of soft-bodied organisms that some of you may be familiar with. This includes the octopus, this includes jellyfish, this includes most species of worm, it includes slugs, it includes plants like moss and climbing vines and oak trees, and it includes fungi as well. That means mushrooms, that means mold, that means slime mold. So soft-bodied organisms are really abundant in the world, and they don't have bones, they don't have shells. How do they even get preserved in the geologic record? And moreover, why should we care whether or not they get preserved? Obviously, a slug or a worm, and I'll concede to you, is not nearly as awesome as a Tyrannosaurus rex. But Tyrannosaurus rex wasn't the entire picture of life on Earth, in fact, for much of Earth history. So I also wanted to show you guys this example here. You've probably heard about germs. You've probably heard about bacteria. You may have heard the term microbe or microorganism or pathogen, things like this. So even if you don't live in a place with a lot of octopi and jellyfish, you definitely live in a place that has tons of bacteria, millions and millions and millions of small microscopic, that means smaller than your eyes can see. You need something to magnify them in order to even look at what shape and size they are. These microscopic organisms dominate the modern world, and they've dominated much of Earth history. This image is actually an image that I collected by camera, digital camera, through a microscope after I had taken some samples of bacteria and washed them with this special dye, this special chemical that sticks to genetic material inside each cell and causes it to glow. So when you take a photograph of these cells under black light, you can see them glow. So every little glowing speck and spot in this image is an individual cell. And you can see I've included a bar up there that says 20 microns or 20 micrometers. So these are really, really small and really, really abundant. And they have no hard parts. That's very important. So why care about organisms that have no hard parts? Well, I will answer that question with a line. So this image contains a timeline from the beginning of the history of the Earth, the beginning of the Earth, to today. You may have made a timeline of some sort during school or maybe seen a timeline. Timelines are very useful. They help us visualize spatially what is actually a march through time. So on the left of the image is the number 4.5 GA. Geologists often abbreviate or say for short GA when they mean billion years. So a 4.5 billion years ago was when the Earth was formed. So in the Q&A after, I can talk a little bit more about how exactly we know the age of the Earth, or indeed how we know the age of any geologic materials. But for now, suffice to say that 4.5 billion years ago, the moon formed, the Earth was this molten ball of rock that is roughly the same mass, roughly the same size and shape as the planet that we live on today. You go a little bit further along, about 4.3 billion years ago, and you start developing the world's oceans. So this process is actually pretty complicated. The Earth's oceans formed by a combination of water, the components of water escaping the rock of which the Earth was made, as well as water being delivered as ice from meteors, from comets, so coming from space to the surface of the Earth. So now we have a rocky planet, we have some water. And as far as we know, as modern humans studying this stuff, the first life didn't really arise on the planet Earth until about 3.9 billion years ago. And the reason that we believe this is kind of complicated. So the Earth is old. And the older a rock is, when you look at it, the more stuff that rock has been through. It's been buried, it's been uplifted, it's been rained on, it's been cooked. It doesn't look the way that it looked when it was first formed 3.9 billion years ago. And it certainly doesn't have any nice hard bones or shells preserved inside of it. Instead, what it has is carbon. There's a lot of carbon in this 3.9 billion year old rock that some researchers found in Greenland. And this carbon was a little bit lighter, a little bit less heavy than we expect a bunch of carbon to be if that carbon got there entirely by abiotic, no life involved methods. So that's maybe not as visually exciting or engaging as a dinosaur skeleton on display in the middle of the museum, but that's pretty profound, that there was some kind of life accumulating this lighter reservoir, this lighter pool of carbon, and we can see that in a 3.9 billion year old rock. Moving along a little bit to 3.7 billion years, and you start to see in the geologic record inside of these old rocks some possible evidence of cell bodies, so shapes made in different kinds of minerals that really resemble the cells that we have today, those little squiggly glowing lines like the bacteria that I showed you in that image earlier. Stepping along a little further, you start to get something called stromatolites. So stromatolites are a huge part of my research personally. And whether or not they contain inside them these shapes that look like individual little cells, they represent a process that requires living things. So if you recall perhaps the last time you had to clean a bathroom or watch your big sibling clean a bathroom, or the last time you went and looked in a pond at the park, sometimes there's this slimy film on some of the surfaces. Sometimes it may stink a little bit, it might be greenish, it might be brownish. These biofilms are made up of millions and millions of those little squiggly lines, those little squiggly bacteria that I showed you in the images before, typically bacteria. I'm simplifying there. There's a lot of different types of microscopic things living in those films. But imagine that you have one of these films at the bottom of the ocean or a pond, something like that, and you have all of this dirt and mud just trickling out of the water, floating down or being eroded in there from a great distance. You have all this sediment that is collecting in that same basin. And the biofilm grows, the sediment collects, the biofilm grows, the sediment collects, and it sticks to the biofilm. It sticks there, and as it sticks there, it might affect the shape of that biofilm. It might affect the size. It might affect what the seafloor looks like. Maybe it starts to look kind of bumpy and wrinkly. You do this for a week, a month, a million years, 10 million years, you start to really accumulate these interesting kind of knobbly shapes. And these shapes, when we look at ancient rocks, we see those shapes in the record. We call them stromatolites, and we try to determine, well, how likely is it that this knobbly, bumpy shape formed because a biofilm was present while the sediment was coming into the basin, or was it some other completely unrelated abiotic process? And it turns out that it tends to be pretty straightforward to tell. There are certain shapes that just don't occur in nature unless you have a living thing there to facilitate it. So the oldest stromatolites, the oldest convincing structures of this sort, don't really show up in the geologic record until between about 3.7 and 3.5 billion years ago. More time passes, you start to see more morphologies of stromatolites, maybe they become more abundant in the geologic record because now we're looking at younger rocks, rocks that are perhaps less processed, less cooked. Things get preserved more often. It could be for a variety of reasons, but we think that it's because perhaps life is becoming more ubiquitous and because the record is getting easier to read the younger and younger we get on this timeline. Little more time passes, you get up to about 2.7 billion years ago, 2.5 billion years ago. We actually start seeing some chemical evidence for life that's really definitive. We start seeing these chemical structures that really remind us of certain molecules, certain materials, certain chemicals that are inside of our own bodies, or inside of the bodies, rather, of organisms that exist today. These steroids, these types of lipids and fats, they tend to be preserved more easily than a lot of other types of molecules, a lot of other types of chemicals inside of our bodies, but they stick around in the geologic record and we're able to find them. So being able to find something that looks a little bit like a lipid in an ancient rock is kind of equivalent to finding something that looks like a femur bone sitting inside of a rock that's maybe 20,000 years old instead of 3 billion years old. So it's a signature. It tells us that a certain type of process is happening. And in fact, this particular discovery, which you all can follow up on the source if you're interested in learning a little bit more about this, but this particular chemical that they found in these rocks tells us that organisms at this time were capable of photosynthesizing. They were capable of taking energy from the light of the sun and some raw materials from the ocean, from the sediments around them, and using it to create biomass, to build cells, and in the process, releasing oxygen. So this is really, really critical. And this is why, when I started this talk, I used the term profoundly earth-shaping, profoundly world-changing. Because once you get oxygen in the atmosphere, once you get oxygen in the oceans even before it accumulates in the atmosphere, you can start creating more and more complex forms of life. And the reasons for this are many-fold, but a simple way of saying it is that oxygen is a really, really good food for living things. Being able to transfer electrons onto something like oxygen means that you can perform increasingly energy consumptive tasks as a living thing. And that allows you to build more complex structures. So a little bit of time passes, and after this advent of oxygenic photosynthesis, that means oxygen-producing photosynthesis, we start to see preserved in the record more complex shapes and sizes of life forms. So these images that I've put up here are some examples of acritarchs. Acritarchs referring to these sort of knobbly individual cells that have little tests and appendages and things sticking out of them. So these are slightly more complex forms. You go a little bit further in time, you start to see individual cells preserved inside of some of those stromatolite structures that I mentioned earlier. You're seeing them together, which is also extremely helpful for confirming our ideas about how those stromatolites formed and how life was involved. This is blown up here, an image that I collected, again, with a camera through a microscope, of some stromatolites I collected from southern Canada that contain some of the best preserved cyanobacterial fossils in the geologic record anywhere on Earth. So cyanobacteria are a very specific kind of bacteria, a single-celled organism that can perform this oxygenic photosynthesis. So being able to not just infer that they were there from the presence of a stromatolite and infer that they had something to do with the rise of atmospheric oxygen or the rise of dissolved oxygen in the oceans, being able to actually see the physical cells associated with these structures is pretty profound. That's kind of what some geologists might call a smoking gun, though of course we have a lot of other processes to weed out before we can say that for sure. This is just a slightly enhanced image looking really, really closely into one of these stromatolites. And you can see there are these kind of thread-like shapes and forms. Those thread-like shapes and forms are the filaments, the cell bodies of those cyanobacteria. So that is a roughly 1.92 billion year old life form that we see preserved exactly the way that it lived and died. So let's see, just reminding ourselves where we are on this timeline of the history of Earth. It has taken us more than half of the entire history of the Earth to finally get some real body fossils, some really convincing fossils, some really convincing evidence of cell bodies. So that's a lot of time where we don't have any hard-bodied organisms, where we only have soft-bodied organisms and they're only very rarely getting preserved. And when they do get preserved, it might not even be their bodies. It might just be these trace chemicals that were in their systems. Stepping along a little further, we have sort of a boring billion where not too much happens with respect to innovations in the geobiological record. But once we get up to about 460, 540 million years ago, we start to see some simple animal fossils. So this is pretty special because these fossils that I'm showing you guys are the first evidence that we have of multicellular life. So that's life that isn't gooey, sticky pond scum made up of lots of little wiggly lines. That's pretty special. Moving along a little bit more, once you get past that 541 million year mark, we are now officially out of the Precambrian. So sometimes when you hear geologists talk about geologic time, instead of saying 4.5 billion years, 2 billion years, several million years, instead of saying that, they might use a name. We've given names to these different periods of geologic history. And so everything before that 541 million year old mark, everything before that we call the Precambrian. And everything after that is the Cambrian, the Devonian, Silurian, all of these things. So you can look up a nice chart that includes all of these names, tells you exactly what they mean pretty easily on the internet. But for my purposes, I think it's simple to talk in terms of ages. So 541 million years onward, we start getting really interesting, complex animal forms, some of which we still see today, or we see things like them on the modern Earth. Other things are completely bonkers. We don't have anything like this on the modern Earth anymore. What is this creature with these spiny, weird legs and a knobbly head? We call it hallucinogenia. Might not even be pronouncing that correct, but it is weird enough that you have to make up these kind of crazy names for them. This is an example of an organism that still exists to some extent on some parts of the Earth. Obviously, this is a much more ancient version of it, but this is a brachiopod. Looks a little bit like a clam or a scallop and other mollusks. Once we get past this Cambrian, Devonian kind of period, we move on and we finally get plants. It took us this long. It took us almost to today. We are almost out of space on this timeline of life on Earth before we even get plants. So once you get plants colonizing the land, you can start to get increasingly complex things going on. You start to get bony fishes. Eventually, plants start migrating up to higher altitudes. They develop more complex ways of performing photosynthesis and performing the work of being a living thing. You get conifers, you get flowering trees. Eventually, you start to get these. You start to get the first flying reptiles. You start to get some of the things that look a little bit more familiar to us, familiar to anyone who's particularly curious about and excited about paleontology. We start to see the big charismatic creatures. I believe this is an ankylosaur, terror bird, all of these big, beautiful, interesting monsters. All of this stuff is really packed in to the very end. Like right before we run out of time, we start to get these complex vertebrate, bony creatures in the water and on land. And of course, I have a little cartoon person to represent ourselves. The way that I've drawn this scale on this timeline, that person doesn't show up until somewhere inside of that little dot that I have to represent today in the modern era. So that means that in all the history of life on Earth, and to our knowledge thus far, all the history of life in the universe, we don't have anything going on between about 4.5 billion years and about 4.3 billion years ago. Then we have this big stretch of time, the vast majority of Earth history, where the only things, the only creatures that we have on Earth are these soft-bodied organisms. So soft, squishy, mostly single-celled or living in accumulations of single cells like those biofilms and those pond scums. But that's most of the history of the Earth. And then only in this last little chunk do you start to get multicellular life. And only a small fraction of that actually has life with real bones and shells and plates, yeah? So if you want to understand the history of life on the planet, then you have to think about these small, soft, not shelly, not bony, not toothy organisms. And that is what I study. And that is what a lot of geobiologists end up studying, because most of the history of our planet involves these creatures that are very small and that are extremely difficult to turn into fossils. Because a question that might arise for you all is, well, is it possible that the reason we don't have more evidence, that we don't have more physical evidence for these interesting multicellular, bony, shelly life forms earlier in the record, could it just be that it's been destroyed because the rocks are old and they've been cooked and they've been mashed up and they've been eroded away? That's certainly a part of it. But there's a lot of different fields of science that come together to tease apart these different reasons for a sparse geologic record. We try to figure out what are all of the things, what's the sequence of events that has to happen in order for a living thing to enter the geologic record. And what about that organism to begin with might make its chances better or worse? And we can use that to figure out in the geologic record the extent to which real scarcity is actually represented. So, as I said before, the record gets a lot sparser as the rocks are processed tectonically, they're buried, they're uplifted, they're cooked, as they're altered by fluids coming in from above and from below. And indeed, as those rocks are colonized by more recent living things. You may have picked up a shell on the beach before and you see all these little holes and bites taken out of it. Animals eat each other. They eat each other's remains. And that continues to happen even after an animal is dead and gets degraded. So all of these processes are other things that we have to contend with. So when we're looking for evidence of ancient life and trying to take from that evidence not just the fact that a living thing was there, but also information about what it was doing, what it ate, how it lived, all of this stuff, we're kind of limited in the tools that we have. We try to look for rocks that are as unprocessed as possible. We try to look for rocks that contain microfossils and microfabrics that are original to the rocks as they formed. And we can work with a tool called stable isotope geochemistry, which we might not have time to get into all of the details of today, but suffice to say that it's studying the chemistry of the rocks to figure out the extent to which living processes and non-living processes have affected the chemistry of that rock over time, even if you don't have any of these physical signals. So microfossils and mineral fabrics are what I'm going to talk about for the remainder of time we have here today. I have this little cartoon video. Imagine we have one of these squiggly little cells. Imagine it's green because it's an oxygenic photosynthesizer. We'll say that this is a cyanobacteria living in the ocean, either today or perhaps billions of years ago. In order to become a fossil, the first thing that has to happen is that that cell has to make it all the way down to the bottom of the ocean. It has to make it down to the bottom of the ocean to get buried. And as it sinks, as it travels through that water column, it has to get there without being eaten up or degraded. It might get eaten up or degraded by a fish or other multicellular creature, if we're talking about that latter third of the geologic record. But for most of history, it's probably likely to get eaten and reprocessed by other microorganisms. But let's say that maybe it makes it to the bottom of the ocean without these processes. Now it's at the seafloor. There's still other things that might eat it and degrade it at the seafloor, multicellular and single cellular organisms. But if that doesn't happen, if we get past that hurdle, then that organism might stay there at the seafloor long enough and intact enough that it can get buried. So these successive generations of sediments, of dirt and mud building up, being shifted by the currents of water over top of it, that has to happen. Then once it's buried, it's still not free, because there's other microorganisms that are living in the soil, in the sediments, and they might chew up and eat parts of this organism, or they might help that organism become mineralized and actually become a fossil. So after this point, if that organism survives its burial, it survives its interactions with all these other microorganisms, and it survives being buried deep and uplifted and eventually discovered by a geologist, then all that's left of it might be a shape kind of like this sort of pale, tiny little squiggle in the rock. And we, as geologists and geobiologists, have to figure out how to go from that pale, tiny, almost imperceptible squiggle in the rock to a whole picture of what the world was like when this organism was living. We have to decide, we have to try to interpret, well, were there other living things also living in that water? Were they performing oxygenic photosynthesis? Was the bottom of the ocean or the lake or wherever this organism was it submerged all the time? Were there currents? Were the direction of the currents changing? Was it an energetic place? Was it a quiescent or calm place? These are all part of the picture of the past that we want to build, part of that story we want to figure out how to tell. So in my work, I often go to the field. I go try to find these rocks that are as well preserved and unprocessed as possible. And I look for these patterns, for these shapes, these wiggly lines that are going to tell me something about what the world was like. So this image here was taken in my field area in Bahia, Brazil, in the northeast of Brazil, a couple of summers ago. And you can see these sort of wavy, swirly, shifting shapes. These are the characteristic forms created by moving currents of water. They're pushing sediments around on the seafloor. They're making dunes. They're making ripples, things like that. So when you see a shape like this in the rock, you get to say, okay, we were probably underwater. There was probably flowing water. There was probably current. It even kind of looks like they go in different directions sometimes. So that current is changing. Where is a place in the modern world where you have currents changing and forming these crests and ripples and swirly shapes? Well, that's a beach. So probably, however old this rock is, that many years ago, in fact, this rock happens to be about 560 million years ago. Wherever this rock was, it was in a beach setting. There was shallow water. There were even storms sometimes that sometimes ripped up pieces of the beach and redeposited them in other places and other piles. So that's important information right there. We know something about what the world was looking like. Because the world where these rocks are today is basically desert and cow pasture. So that's a pretty profound and awesome revelation that the world of northeastern Brazil has gone from being this underwater, highly energetic beach environment to being a cow pasture. That's pretty cool. So, in addition to looking for these forms that tell us something about the physical processes, we also want to look for forms that tell us about the biological processes, the biochemical processes. So here's an image also from Brazil that involves these sort of blobs and bumps and things. Remember, I was talking earlier with the stromatolites about how biofilms, as they accumulate sediment, can sometimes change the shape of the seafloor. This is a possible example of that. So nearby, in that very same picture, you also start to find stromatolites. Those stromatolites are sort of stretched out in this plan view. So imagine you're standing on the seafloor and looking down, or maybe you're snorkeling and looking down at the seafloor. This is what you would have seen roughly 540, 550 million years ago if you were standing where those rocks were forming. So after I go to the field and I collect all of these samples and I make all of these detailed sketches and images, I bring those rocks back to the lab. We cut them. We cut them with a diamond edge saw because they're pretty hard rocks. And we shave them, we grind them down into very, very thin pieces that if you hold them up to the light passes through them a little bit. And the way that that light interacts with the materials that the rock is made of, it allows us to see details that we couldn't just see when we were looking with our naked eyes at the rock in the field. So it's important to see these larger textures in the field and to have that context because it tells us a little bit about the physical world. But we have to dive in deeper and look on this micro scale in order to look for evidence of not just the presence of life, but the style of life, the habit, the method of surviving that these organisms that built these structures had. So in this process, I take lots and lots of pictures. I make lots and lots of annotations. I compare the pictures that I take from samples in different parts of the same region or across the entire continent of Brazil. I try to reconstruct a picture. I do a lot of drawing and sketching to this end, where I try to understand what was it like to stand on the seafloor, what was it like to be a living thing in this world 550, 540 million years ago. So that's microfossils, mineralogy, mineral fabrics. Stable isotope geochemistry, as I mentioned before, is another tool because sometimes when I get those rocks cut, grind them down, look at them through the microscope, sometimes you don't really see nice, pretty shapes. We don't see the original grains. We don't see anything that looks like a fossil. And in those cases, there may not be a lot more that these rocks can tell us visually. So instead, we turn to other methods where we dissolve the rock in acid and we try to collect the gases that come off of that reaction. And we measure how heavy or how light they are. This is something I mentioned earlier, and something that we'll probably have to get into in more detail in the Q&A. But measuring how heavy or how light these different rocks are when we put them through these chemical processes, that also helps us to figure out what kinds of living things might have been there, even if we can't see them. So why do we do this? What can we learn from this evidence? So I've talked a little bit about how we can learn what the world looked like. Are we on a beach? Are we in the deepest part of the ocean? Are we up on the top of a mountain? What was the world like when this rock was formed, when this thing was living there? But in addition to that, we can get information about what the organisms were eating. What were they excreting? And that tells us something about how were these organisms interacting with and shaping and changing the world around them. So we can walk outside and we can see very easily every day in our communities how living things are changing and shaping the world around them. We know that when a dog digs a hole in the garden or in the park, then suddenly you have a place where water and other sediments and maybe trash and things can collect inside of that hole. Maybe a seed will blow there from somewhere else and it will land and it will push more sediment out of the way. This is a small thing that may not enter into the geologic record, but if it did, if that hole was preserved, if there was no other way for that hole to form other than the dog digging the hole and all this stuff, that's really important and interesting information for a future geologist millions of years from now, trying to figure out what the world was like. The world had dogs, the world had trees, the world had soil, and those dogs, the trees, the soil, they were all interacting with one another and shaping each other. So taking this information, we can move forward instead of looking back, and we can start to build models and predictions of how organisms now will change the environment for the future, and vice versa, how the environment will shape and determine how organisms behave. So in some ways, the study of ancient life, past life, is a purely intellectual, purely curiosity and creativity-driven exercise. But it can also be an incredibly useful exercise for figuring out what the world will be like in the future and how we can prepare for it. So the tools that we use to do this are myriad. I've included a few fun pictures here. Whether you enjoy drawing and painting, spending time in the lab, whether you enjoy horseback riding and hiking and hanging out with your friends, all of these things are part of the process of being a scientist who studies ancient life on Earth. And that's one of the things that originally drew me to the field and that keeps me in it now, is that really the best way to do this science is with a lot of different types of creative people working together. So with that, I will put up a slide with some resources if you're curious in learning more about the history of boneless, shellless, soft-bodied life, and in fact, microbial life. Two of these books were written by professors of mine that I've both worked with and taken classes with in the course of my educational history, Joe Kirschvink and Andy Knoll. The last book, Strange Creatures, exists tattered and much loved on a shelf at my parents' home back east. It is one of the first books I ever had and an excellent, really awesome look into the history of complex multicellular life on Earth that will hopefully get you excited about some of the aspects of paleontology that maybe don't involve dinosaur bones. Thank you guys so much for joining us here in virtual Beckman Auditorium.
Thank you, Cecilia, for your fascinating presentation. Our website has a link where you can ask Cecilia questions and she will respond within 48 hours after the initial presentation of her lecture. I also want to thank Mitch Aiken and Kitty Cahalan of Caltech's Teaching, Learning, and Outreach Office, as well as Mary Herrera from the Office of Public Programming, and the entire academic media technology team and the team from Event Productions. And thank you to the Caltech Employees Federal Credit Union, the Friends of Beckman Auditorium, and our many sponsors who make these types of programs possible. We look forward to your joining us again for other performing arts events and science lectures, which you can find out about by checking our website, events.caltech.edu. Thank you for joining us.